Then we move on to the next section here, RNA-seq. So how are we? We're a bit behind. Here we're going to have a look at import of BAM files, how to add annotation and save as a GE data file, and then once you have loaded your data, how you can analyze it. So we close down this data set. Again, from the log tab, we just close the data set. Now I will want to load some BAM files. So I do open BAM files. Then I must have the reference genome for that corresponds to the reference genome that was used for the alignment. I have the possibility to do a pre-filtering here. I can select my normalization method. We have 3 to choose from. TMM was the first one that we implemented. And we added the other ones because there is a request to have more normalization methods. And you can also set if you want to work with stranded or non-stranded DNA. Then you select your files. So I pick, let's say I pick all of these 4 very small BAM files. I selected them and then I press OK. So now what we are doing here is that we are counting the BAM files, creating a data matrix over the counts, how many reads you have per gene. Then we will do the normalization, and then we will do the log transformation. And we will display a QC report giving you some more information. So this is what we see here. So here we have the base position quality, showing that one sample is very okay and one sample is not so okay. And we have Spearman and Pearson correlation here. And when I close this, here I have my 4 samples. And we have, as you can see, 60,000 genes in this data set. Okay, good. And then I don't want to do this normalization again. Now it was very fast, but in normal life, you would have BAM files that are 1,000 times bigger. So then it will take a couple of minutes per BAM file to import. At least 1 minute per BAM file, or maybe up to 4 or so, depending on your processor and so on. So once you have done this, you want to do save as a GE data file. Save as GE data file. The next time you want to open this data set, you can directly open the normalized data set. Okay, and then we would like to do some analysis, but when I look at the annotations that I have here, I don't have anything that groups the samples, so I need to add annotations. So either I already have the annotations in another file that I can just import, or I want to create the file from the software. So let's have a look at that. We do File, Export, Sample Annotations. Create an annotation file, put it on the desktop. Here we are. I open it in Excel. It's a TXT file. The good thing about doing it from the software is that you then get the unique ID correct. If you want to import it from another file, you must make sure that the unique ID matches whatever you have imported. So here we have it. So now I can just add annotations here. So I can say sex, for instance, and treatment. That. So here we have female, male, male, and here we have control, treated, like that. Then I can add as many columns I want here. I save it. Then we go back to the software and say, now I want to import. So import sample annotations. which file? Let's find the file. Here it is. So now we have sex and treatment here as well. So press OK. Next time I look here, you see that I have sex. Okay, that does not seem to be a very strong signal. Treatment seems to be a very strong signal, as you can see. And then we could do an analysis. We can t-test on treatment, for instance. Control versus treated, or the other way around. You can see that, yeah, we find 60,000 of the genes that are really, really good, a good separation for the two groups. And then again, I want to save this as a GE data file so that my annotations are also in the data set. Let's continue. So here we have more details regarding the import, and there are, as I said, 3 different normalization methods. We don't recommend which one you should use. They are all okay. When we started, we only have one, which we know is very. So if you are in doubt, you don't know what to use, you can safely use TMM. So you should have your own opinion on what normalization method you would like to use, but TMM definitely. But there is a lot of documentation, of course, on the web regarding this, different opinions, depending on what data set you have and so on. And here is some more information on how to add the annotations. You can try this, and we will try this in the advanced No, I actually did not include that in the advanced training. So you need to try this on your own. More options. If you have very many BAM files, and or you maybe have a pre-normalized data matrix, you can import it via the wizard. Then somebody else already did the normalization for you. And if you have a count matrix, you can also import that, and we will perform the normalization. A count matrix is a data result file that is not normalized. It's just a raw counts from the RNA-seq analysis. And I will show you how we do that. So let's just close down what we have open here. And you will go here, open or open with wizard. Find your raw counts dataset. Here I have a count data matrix. And in this case, yes, this is a raw count matrix. So here are the raw counts. But when we normalize, we want to take into account the gene length and yeah, at least the gene length here. Sometimes also the read depth. So yes. Here again, you can select one of the 3 normalization methods. So, and it asks, and we also need then to include the feature length, the gene length here, because we need that in the normalization. So it should either be in this file here as a variable annotation or in a separate file. In this case, it's in the file here. Next, select separator, tab, variables are rows, yes. Top left cell, yes. Top right cell. Next, variable annotations, and you need to also include this one, include it. Next, and then you pick the sample annotations. And in a last step here, you need to confirm which is the gene length. It's this one. Then that brings in the data. And if we look here in the table view, you see that here we have the normalized and log transformed values. And then, of course, you save this as a GE data file. So you can quickly open it the next time. So that's a new nice feature. Okay, any questions? on this? No, then I continue. Thank you. Then we go to the, okay, so there are some questions here. So let's open up the chat again. I'm not sure if the data that you describe here, if it's any different from other. I can take this question and check with my expert colleague. So I'm just making a note of that. I guess you would then have that as a count matrix, or so I'll just make this. I might need some more information on you. Maybe you can send me an email with more information regarding that question, and I can take it to my experts. Thank you, Laurie, for participating. Yes, it depends on your, I would say, on your reference genome. So I think so this is information that if your platform is performing the analysis for you and creating the count matrix, this is information that they should have. I can check that as well, where to find gene length. Okay, so Simon, I got a reply from you. Okay, I'll try to find this out and contact you separately after in emails. Okay, then we move on. Now we will talk a little bit about analyzing single cell data. So here we will use a real data set made publicly available by 10x Genomics. It contains 2,700 cells from human peripheral blood. And what we are going to have a look at here is to findings, subpopulations, cell subpopulations. And to do that, we will use variance filtration, taking away some of the noise in the data set. We will visualize using PCA and t-SNE, and we will also color by gene or variable expression to help us find subgroups. And the last thing we will have a look at is how to subsample a big data set in order to make it a smaller, so that it becomes more interactive. If the data set is too big, it can be very slow to work with, frustrating. So then you might want to make it a bit smaller. There is more information on this in the help in the reference manual when you search for t-SNE, subsampling, and so on. And there is also a recorded webinar on analyzing single-cell RNA-seq data. So let's have a look at this. I close down this data set like that. And then I will open up a single-cell data set. This one. This is already saved as a GE data file, and it has been pre-normalized. So here we are. So when you see a data set that looks like this, you need to do something in order to make it open up, because here we have, as you can see, 2,700, 2,600 samples and 13,000 genes. And the only thing we can see so far is that these cells here are different from the rest, but there are probably a lot of more information in here. So let's try to filter by variance or standard deviation to take away genes that don't vary so much and see if that can open up the data set. So I move the variance slider here, start filtering. It's quite a lot of data here, so I know in this example I know where I want to go, so I just type that in. So what I did now was that I filtered away around 4,000 of these genes. So here I have filtered away, say 25% or so of the genes, and that really makes the data set open up, as you can see. So it looks like I can see 3 or even maybe 4 groups here in this PCA plot. So I can assign the groups here if I want to, but I will actually move directly to a t-SNE plot, 2D t-SNE, which is a very popular plot in when you work with single cell data, because the t-SNE plot actively clusters samples that it believes belong together. Which makes it a bit biased, but very useful. So you should actually try a couple of perplexities and see. The perplexity is your guess on how big the clusters are. So try a couple of perplexities to see that you get a stable outcome. And since you already, so I'm testing 50, I had 100 before, I'm now testing 200. But we started with the PCA plot, and there we got a good impression that we could probably find 4 groups in this data set. And the t-SNE plot actually confirms that. So by using it together with a PCA plot, you have a good idea that you are in the right area. So all of these 3 settings that I tested actually gave me 4 groups as a result. So that really confirmed what I saw in the PCA plot. But as you can see, the groups are much more separated. So what we will do here now is that we will save these groups, because so far we see that there are different groupings, but we have no idea what they are. So let's create a new annotation and give it 3 more values. And then we use the annotate mouse tool to say that these are the yellow samples. Color by that as well. These are the pink samples. These are the white samples. Okay, goodbye, Usma. I saw your message. Thank you. Yeah, I'm running a bit late. I'm sorry for that. Okay, so this is one way of finding possible subpopulations here. There could, of course, be more information here. So we have, there could maybe be a group, here could maybe be a group. So let's now try something else. Let's color by biomarkers for specific cell types. So I know, for instance, that B cells are, you have a high, so I'm looking for CD79. So I'm typing CD7 very quickly here. Trying to at least. So this is where I should be. So CD79. Here we are. I expect upregulation for CD79 for B cells. So I color by that using this color tool for the selected gene. Then you see that, okay, I have a very clear upregulation in this group. This is probably B cells. Let's try the same for CD79B. Maybe not as clear. I have some upregulation here as well, but it confirms the results. So this could be a good way of understanding what the different groups are. Then I have a marker for natural killer cells. So let's try that. That's GNLY, I think. Here we are, that one. Let's try that one. Okay, so it looks like part of this big group could be NK cells. Interesting. To get a clearer picture, I'll take away all the other groups here. That one as well. And then we will do this again. Then it's easier to see. So I'm now just focusing on this group. So then I could maybe try to annotate this group as well, exactly where I want to. It's a bit hard, but something like that. Did I manage? Yes. And then I can put back everything again. And then, so the t-SNE needs to be manually updated, as you can see. Here we are, and then maybe we want to color by our new annotations. So this is one way of working with single cell data. The last thing that I'm going to show you now is how to subsample a data set. So let's say that this data set was so big that it was a bit slow in updating and so on. So then maybe I want to create a smaller data set in order to make it easier to work with. So then I choose subsample from the file menu. And then I can stratify based on things that I know. In this case, there were actually some annotations, but let's say that the only thing I had was my new annotation. So then I want to create a smaller data set that's maybe 50% of the size of this one. But I want the proportion of the groups that I've identified here to be the same in the new data set. So that's what the stratification means. Then we press OK. Then we are creating a smaller data set, a subsampled. Let's put that on the desktop. That. So here we are. This is the new data set with half the number of samples. So if we do a variance filtration here as well, and then maybe color by our new annotation, we see that, yeah, we have the same groups in here, and it looks like they have around the same proportion, or they have the same proportion as we saw earlier. So that's one very good thing. You can also subsample without any stratification. You can just do a random subsampling of your data set just to get it down in size. And then let's have a look at the last slide. Ways to import single cell data. Since you have thousands of samples when you work with single cell data, importing BAM files is normally not a very good idea because then it will take too long. If you have hundreds of samples, you can definitely do an import and normalization overnight. But when you have thousands of samples, then you will block the software for too long. So then we recommend that you work with either a pre-normalized data matrix that you just bring in via the wizard, or a count matrix that you normalize, as I showed you before. And then we have one other option. We have created a template for importing 10x genomics data. So here it is in the template browser that you find here. There is a template written in Python for how to download 10x genomics data. So it must be from the Cell Ranger pipeline, count or agr. And here are some instructions on how the setup. So this will import and normalize 10x genomics data. And just a comment on the template. So these are workflows that we have created in the software written in Python, and they can be automated to, for instance, download things, or just to perform testing and pull up plots and so on. We also have a template for downloading TCGA data, for instance. If you are fluent in Python, you can actually make your own templates as well, because the scripts for these templates are included together with an extensive manual. So you have a lot of options here if you know Python. Otherwise, you can just use the templates that we have created. Yes, I think that was what I was planning to show you. Thank you very much for attending, and I will now open the chat again. So any more questions? Questions on single cell? No, I don't see any questions. So I just want to remind you, if you plan to also join the advanced training tomorrow at 1 p.m., we now know that the best way to do this is to register on the home page, because the webinar is posted there, and then you will get the meeting link sent out directly from WebEx. So I will send out an instruction regarding this again, since I actually sent out the wrong instruction earlier. But all of you who attended today knows how to do that, so that's good. So if there are no, or are there maybe, is there maybe a question? Okay. Can you, one question, can I do, okay, Annika, thank you very much. Speak to you tomorrow. Xinlei, can you do GSEA using single cell RNA-seq data? Yes, of course. As long as you have imported the data set in the software, you can do anything on it. Then you just analyze it in the normal way, and you can also perform gene set enrichment analysis on it. The only thing that's important is that the unique ID matches the pathways that you want to compare with. Another question is, where can I download databases for gene set enrichment analysis, for instance, KEGG pathway? So it's not the databases that you download, it's the gene sets. So it's the actual pathways that you want to download. So you need so as I mentioned here in the beginning, there are a couple of data repositories, for instance, MSigDB, Molecular Signature Database from Broad Institute, Reactome, Gene Ontology, KEGS, and so on. So you visit these repositories and you search for pathways or GMT and find files that you want to download. Great. So people that has to leave, thank you for today and see you tomorrow. And for the rest of you, any more questions? And I will send out the slides to all of you. Great. I keep the chat open for a few more minutes, but for your time today and for you that are joining together, I look forward to speaking to you then.